Welcome, fellow beekeepers. This is Jamie Walters, OSBA's Maumee Valley Director and News Editor. I'm pleased to announce OSBA's new live webinar training series. Live sessions are recorded on the second and fourth Sunday of each month, 7 p.m. through Zoom conference call. You're welcome to attend by making a reservation on OSBA's website at www.ohiostatebeekeepers.org. Go to the events page and scroll down to the event you would like to attend. Confirm your RSVP by filling out the registration form with your city, state, name, and email address. Approximately 2 hours before the beginning of Sunday's webinar, an active Zoom link will be sent to your email address. Each training session will be recorded and posted on the following Sunday on OSBA's new YouTube channel. You can find it at Ohio State Beekeepers Association. You're welcome to subscribe and be sure to click the bell to be alerted each time we upload a new video. Each month's speakers and presentation will be posted on the Facebook page, by direct email to registered and paid members, and our website at ohiostatebeekeepers.org. If you have any questions, please feel free to contact me at editor@ohiostatebeekeepers.org. And now on to our previous recorded webinar.
Thank you, Jamie. And Ohio has great credentials for beekeeping. We get a ton of samples from Ohio for the Diagnostic Service, and it's not because your bees are terrible. It's because there's a lot of really active beekeepers, really engaged beekeepers at every level. So, and I also, of course, know many of the inspectors and folks working there as well. And while I haven't met Jamie in person, I know his name quite well, and I'm very honored to talk to all of you. And thanks to him for setting this all up, doing all the hard work. I will try to keep an eye on the chat screen. I'm trying to see if it shows up while I'm sharing, but if I miss it, I'll get those at the end. But if you do have questions, I really don't mind if you unmute and ask them as I go. That's totally fine. And I promise not to take an hour of PowerPointing you to death. I'm just going to kind of step through different projects at the lab right now, and some of the issues, some of the challenges that many of us as beekeepers are facing, but also some research angles to try to address those. And then I'll end with a reminder review of the Diagnostic Service. So the Bee Disease Diagnostic Service is run from our laboratory, and it's a free service that is a little bit changed in current days because of staffing challenges as we get back to physical presence on campus, but it's still alive. So I'll start there so it doesn't, I don't take too much more of your time. Our focus as a laboratory and in this area, starting with the first kind of bee research in Washington, D.C., and then moving out to Beltsville, where we are now in the late 1930s, is on honeybee disease. So our real focus has been, starting with the foul broods, and as mites arrive, tackling some mite issues, trying to address mites, and also tracheal mites and Varroa. And we actually do some work on the still exotic mites that could well arrive here, and try to preemptively look at some controls for those. But I will also highlight, we have a couple scientists doing things that are distant from disease, although we work together and we try to, pretty much any experiment that's run in the field in the laboratory here, we'll throw in a couple of disease checks just to see it as a marker, if nothing else, of how well the bees are doing, or how badly they're doing if it's, say, a chemical stress experiment. So I will go on. This is our group from a couple of years ago. We have 5 scientists working with honeybees directly, some students and some great technicians who are helping as beekeepers and in the laboratory. And then about almost half of the lab, at least in the summer, are visitors or short-time students coming through to help with field projects and in the lab from all over the world. So we have a pretty international bunch that comes through. This is the hardest slide of the day, but I just wanted to say, and the main point here is we're looking at disease diagnostics, also treatments, new treatments for disease. But our real integration with the lab right now is to try to look at things like overwintering, where we know we lose a lot of bees, build up in the spring, and queen quality. So in a sense, our lab is we're not inventing better bees. There's the USDA laboratory in Baton Rouge, Louisiana, that's their focus is on breeding bees and testing stocks. We take some pretty generic honeybees for most of our experiments, and as you might know them, the sort of mass-produced honeybees from the southeast for some of these experiments, because we want to stress them out. We don't want bees that are at that point especially adept at mites, for example, because we're doing a lot of work on mites themselves and on the bees that are suffering from mite disease, Varroa disease. So we do have, I'm happy to say, some colonies that are actually from West Virginia, so the bridge state between our two, I guess, and some more resistant bees, and those are nice to work with. We're having some success with them, but our real strategy isn't to get a super bee. It's to keep almost any bee alive as long as possible. And as beekeepers, even if you started a couple packages this spring and are watching them, and some work, some don't, or some in your club have success and some don't. There's a real challenge when things in a beehive fall apart, and it could come from a queen loss event that isn't fixed by the bees or the beekeeper. It could come from starvation. Even now, as the flow tails off, there are colonies that maybe never quite built up, never collected much, or the beekeeper took a bit too much of the goods from them. And so there could be starvation events and general disease. You were talking about small hive beetle earlier, and that's sure a sure sign of a colony that's not quite right. It's not strong enough to fend them off, and it can go downhill in a hurry at the apiary level, and that, not to mention the foul broods, which can do so. This slide you've probably seen or versions of it. This is actually generated by our neighbors here at the University of Maryland and the Bee Informed Partnership, and many of you, because you are contributors to this survey each year. And the general rates of total losses in colonies and winter losses are holding pretty steady, and they're steadily quite high. 40 in some years, approaching 50% losses annually to beehives, and that is a tough thing to continue a hobby with or to make money from, because you're constantly splitting and replacing those colonies or buying new colonies in some cases. The little bar at the bottom is kind of from beekeepers who have been in the business for a long time. That might be their comfort zone. They, post Varroa, if we could get back to 15% losses, things would be more sustainable. Well, the short answer was I was saying that things aren't so great in terms of colony losses, and at the same time we're finding success, or beekeepers are finding success in some years in some states. Ohio's right here. I don't know if the cursor's working. Middle of the road, this is for 2018 to 19. Better at least than the samples or survey results coming in that year from Louisiana and Nevada. But in general, the upper Midwest and then moving towards the East and the Mid-Atlantic faces these sort of 50+% losses every single year. And that's what we, or beekeepers, are trying to get around, to try to get things back, to cut that by 2/3 or so. So it's not such a difficult prospect to keep your bees alive. This is from one of our research apiaries, actually one I had set up to do some projects last year. And it was another, every year is a lesson with bees sometimes. And these colonies stayed healthy all summer long. And we just they started with packages, again, just kind of generic packages from certain mass producer in the Southeast. And they did okay though. They built some mite numbers up, which was good because we wanted some virus stress on them. But our virus stress built up too much. And over a matter of weeks, we saw something like a parasitic mite syndrome. We're not quite sure, but the virus levels we know were quite high. Workers were not surviving, as long as they should. It seemed like maybe they were disappearing soon after becoming foragers, so 2-3 weeks old. They weren't doing well. And very soon after that, the brood patterns got really spotty and really messy. And this entire apiary actually fell apart over a matter of weeks. It wasn't the not so mythical, the formerly more observed colony collapse disorder, in that, there weren't these nice, clean brood patterns and no adult bees. There was sort of spotty brood and other bad signs as these colonies started to fall apart. They did have queens though, and they had eggs, but there was something very bad going on both during development and in the adult bees by that point. So what is going on? Well, I'd like to highlight 3 of the research directions at the lab and talk about the 3 main stresses facing your colonies. One is chemical stress, and this is a new project or new program in the lab in the last couple years. Dr. Mohammed Al-Baraki, he's a long-term beekeeping family starting in Syria, but he grew up in Canada, in Montreal actually. He knows his bees incredibly well, and he knows the economics of beekeeping from his father, and kind of keeps that in the back of his mind. But he's also looking at kind of the physiological, the bee level stress of pesticides. And the long-term goal is queen survivorship and fecundity. So keeping queens themselves, surviving more than just a season, ideally a couple, but also, producing healthy and consistent egg rates, so brood patterns consequently from that. He and his group are now, they're looking at some of the diversity of bees that we have. And again, I mentioned at the start, if you could hear it, that we have mostly, I have to say, mostly kind of Georgia Italian queens, but we've brought in stock over the years, even some Russian bees in years past, and then more recently some bees from West Virginia that are they're actually they're arguably still an Italian origin for the bees, but they've been put under some mite selection for a while, and they're a bit different. They look a lot different than the ones we had previously. So he's kind of establishing the diversity in our apiary. He's also very interested in kind of landscape questions, focusing for now on the stress of those landscapes in the form of chemical stress and how that impacts bee health. This is one chemical mineral that he's worked with, and I just show this because it's sort of a typical test that you start with in these sorts of projects, and it's not very realistic from the bee standpoint. They're not going to flowers, they're not forced to fly, but of course you get very direct views of relative stress and relative impact of different single chemicals on bees. And that's about as far as you can get with those sorts of studies. The next stage, and this is a really exciting phase of research for bee research right now, is to tag bees and actually follow them over their entire lifetimes. And that's what Mohammed is up to right now. And he's following some studies, this one in particular, where the researchers put these tiny tags onto each bee, and these are the same tags that are used for stock control in stores and things like that. They're close range tags or reporters on the bees. These won't work, to watch a bee flying past in the sky like a transponder or something that's really stronger. They're not energized at all, in fact. But as this bee travels past a scanner as she comes back to her hive, that scanner will tell exactly which bee came in and which bee went out, and it can handle thousands of data points. And these things can be coupled with microbalances, little scales that the bees light onto when they land and take off. And you can actually measure the foraging success of individual bees, as well as the number of trips they're taking. And this is a very typical graph of what researchers find with these sorts of data. In this particular case, it's with imidacloprid, which is one of the pesticides, but you could interchange many chemical or virus stresses and get the same result. And what effectively happens is this purple curve is these are from the bees that were given a dose of this at birth as larvae, actually, so before they're flying or even crawling. And that dose will lead them to drop out of the picture at a slightly higher rate throughout their whole life. And it looks very subtle, right? It's the purple line below the green line, but that's enough to tip a colony from being a growing, vibrant colony to one that's in decline. All it takes is this kind of steady drain of bees falling out of the system. Especially if they're unable to forage, if they've been invested in and they're gotten all these resources from the colony, but they never really go on a good foraging trip. That's really costly for the colony, and it can be subtle, but actually end up bringing down the whole colony. So generally speaking, he's trying to look at the queen health, but also these colony traits. And then just to see, and the goal isn't, we can't find too many bubbles where bees are not exposed to chemicals, but we can get the relative stress of different ones and then work with growers to kind of, help decrease that stress. Food stress and nutritional stress is another angle that we're working on in the lab. In this case, it's Dr. Miguel Corona. And he's also doing a lot of individual, these sort of nucleus colony studies, especially looking at particular diets that may help seasonally, maybe going into winter to help bees make it through winter. And this is one recent study from his group where these were actually commercial bees, traveling bees in Uruguay, but we did the lab work or the sort of chemical analyses and such here with a student who worked with him and then was a student down there. And this was showing basically that the diversity of pollens that were observed on these bees was tracked very well with both brood and adult bee populations. So it was a hint that these sort of monofloral or monotypic pollens are nutritious. The bees can get by on them, but they do a lot better with a range of different pollens. And a lot of Miguel's work right now is looking at how that may be supplemented either by the beekeeper in terms of a pollen or protein supplement, and also maybe how to look at kind of seasonal changes in forage that might really help the bees. To get back to the disease, to kind of switching back to the disease front, these bees also, the ones with this monotypic pollen, also showed higher levels of Nosema, the parasite Nosema. And so there are signs that the sort of interplay between what you eat and the parasites that might take advantage of bees. So this might lead to a new diet supplement. In the short term, it could lead to recommendations, some of which are intuitive to the many beekeepers are gardeners or master gardeners and know their stuff when it comes to flowers. And I know well that many of you have thought deeply about this, but the general recommendations would be to kind of keep your bees on a diverse at least pollen flow, if not nectar, and also to try to if they've been on a single crop or a single planting for a long time, maybe there will be a time when you could supplement them to get them kind of the missing elements of their diet. So more bad news, I guess, for the country. These are numbers for deformed wing virus, which is the most common historically virus in bees in the US. And again, almost every colony in well-surveyed states has this virus. It's super prevalent. It causes deformed wings when at really high levels, but it also causes these kind of low-grade or these increases in mortality, the middle-aged bees that are dying in the colony. And so any amount of it is bad news. We had a hint last year that this virus was going down. It wasn't quite sustained through the year like these levels. It was decreased from the 10 years before this particular virus might have been going down. And that seemed like great news. We thought, well, maybe there's resistant bees or they're getting better forage. But at the exact same time, this virus, which is a very close relative, showed up and just started filling bees. It's sort of taken over the top spot, as it were, as a bee virus. And it's equally bad for honeybees, unfortunately. This graph shows, the first signs of it were in 2010, just a couple positives out of 75 colonies across the states that were surveyed that year. And then in 2016, it had already become almost as common as the traditional deformed wing virus, and now it's more common. So this is a case of a transition in real time from one virus in bees to another. We think this one came from Europe. Actually, it's been a problem in Europe, and it's associated with Varroa mites there. And so the mystery is how it did get here. But clearly, once it arrived, it did really well and took off. So what's some good news possibly? Well, one thing is we can target these viruses. You may have heard this, everything from, well, good nutrition in general does seem to reduce virus loads. People have tried, and we've collaborated on mushroom extracts. We've collaborated on nectar compounds. In this paper, this is sort of describing some studies with nectar compounds. And then even some of some potential drugs would be the real, like the medical antivirals, or the ones that aren't used, of course, for medicine, because we don't want to compete with those. So there's some promise that we can find antivirals in nature. We're tackling a project that would be in full swing this summer, but for another virus, trying to target viruses directly in bees with these natural products. And the natural products angle is partly pragmatic. The regulatory horizon, the length of time it takes to get these on the market is much less than another veterinary drug, for example. So it would be easier to convince someone to kind of push it to market, or maybe for beekeepers to just simply use it on their own if it's labeled and tested appropriately. So that's one angle we're trying. We did get about 45 or so done last year. This year's been sort of a bust so far, but we might sneak some through at the end of the year when another project here finishes up, and partly because again, a lot of this can be done in the laboratory. We are screening, let's see, this is a close up. That was a happy summer team a couple years ago. Our first screening, like the pesticide testing, can be done in these cups. These are 30B cups, and they get sugar water. Believe it or not, they'll last in an incubator for weeks like this without any pollen or any protein source. Once they're old enough and their guts are established, they'll survive on sugar water. And so we know they're carrying virus because they're from colonies with this virus. And we in their sugar water sort of IV drip, which doesn't drip in their bodies but into their mouths, we can put low levels of these natural products and try to just measure their virus levels over time. This is Lotmaria, which is a trypanosome parasite, sort of the parasite formerly known as Grathidia mellificae, and Grathidia got replaced in ecological space. So the new sister species of it, or new genus actually, of this parasite is called Lotmaria. And these are them. They're growing in the gut. They grow in the, basically in an offshoot of the small intestine or the ileum of honeybees. And they compete a bit for nutrients. In bumblebees, they actually really affect overwintering survival of queens, and they also shorten the worker lifespan. In honeybees, it's hard. Nobody's seen a sort of colony level effect of them, but they're a parasite. They're not good for bees, and so what we're trying to sort out is whether we can take this parasite growing in cells, and that's growing in basically food. It's not inside a bee at all. And again, use that system as a way to test medicines and look. They're gut parasites that we think are affecting honeybee health, and we're sort of just getting a handle for how common they are. There was a survey, once we sort of named this new species or new genus, and it was found in Spain by a lab that's looked at a lot of Nosema effects on honeybees. And it may be sort of at the level of Nosema, where it's not going to take down your colony absolutely, but it's an energetic drain that is yet another thing that you don't need to have your colonies facing. So we're looking at that. There may be a nutritional fix to it. There may be other genetic resistance. So trying to find out if that parasite is really treatable, or if it's better to sort of breed in resistance for it. I just wanted to highlight, this is a project, a side project for me, but it involves some Ohioans. So I wanted to highlight this as a, and we talked to some folks in the state about mostly disease and like what you might use for diagnostics. I know we've talked to some of the inspectors about this. Like they go to when an inspector comes to your colony, they will take a very holistic view, starting with meeting you in the first place as a beekeeper and asking you questions and your observations of what happened or didn't happen to your colonies. They'll certainly do a visual inspection of the colony and the surroundings and the stores and the brood and the adult bees and the mite levels, and sometimes Nosema, and then make an assessment on all these components as to what either went wrong with the colony or what's at risk there. What we're trying to figure out is if there are other field diagnostics that might help an inspector or a beekeeper sort this out. And this was the first sort of stage of this. We did a lot of interviews of folks in bees. We've now talked to 130 or so different beekeepers and inspectors nationwide. And maybe I don't think these were loaded questions from a disease group, but a lot of the concerns were over health and disease in particular. Certainly, pesticides were mentioned and were a factor, but there's a consensus of sorts that bee disease is still a factor in your colonies. And the differences between many of you I know are small scale beekeepers, trying to deeply attached to your bees and willing to open them and check on them a lot, but also basically trying to get a handful of colonies through each winter. Commercial beekeepers are doing the same at a much greater scale, but they're also very concerned about labor, right? They have trouble retaining a labor force. They're not getting much of a margin on honey these days, so they're concerned with strategies that might that would decrease the amount of time you have to spend in your colonies, as it were. I know this sounds terrible to a passionate beekeeper, because you want to spend as much time as you can in your colonies, but if you're doing it commercially, you can't afford to spend even 2 minutes on each hive and each visit. You've got to be in and out really quickly. So we've talked to a lot of folks, and we're trying to compile the insights from everyone. And again, the short answer on the diagnostic side, at least, is if we can get something that you could use in the field for viruses, it would help in terms of giving you one more bit of information, I think, for that. Okay, so let me now move to the last scientist in the laboratory currently who I'd like to talk about, and that's Dr. Stephen Cook. And his work is kind of connecting, and sorry, Dr. Judy Chen as well, but will come later. But he is connecting the virus expertise in the lab and Varroa. So trying especially to look at the impacts of Varroa on bees in terms of physical stress on the bees, as well as virus transmission, and then also look at how that interacts with other things like pesticides and temperature stress. Right now, his focus is on a foundation grant that's a large team, including Reed Johnson, who's in your state, great bee research professor, researcher, and teacher there at Ohio State, and others trying to find new and sort of available chemistries that might control Varroa mites. And this is the strategy for that. Again, all of these experiments start pretty small. You either put mites into a vial in the lab, or you put bees into a cup in the lab, and that's kind of the strategy early on for much of research, is to reduce it down a bit to these pretty small scale things. And for each chemical that's tested against mites, there has to be a much lower sensitivity to that chemical with the bees that are exposed to it. So it's always hand in hand. You want to get something that kills the mite on top of the bee, but doesn't hurt the bee nearly that severely. And there are some compounds that do that, some of which have been on the market for a while and are maybe losing efficacy because of resistance by the mites. But that's the goal, is to find a handful of new compounds that are hard on the mites but not so hard on bees. Right now, they're actually doing, in fact, in West Virginia again, we have good collaborations with beekeepers in West Virginia. They are doing some field testing on some of these compounds. So they're trying to see if these are working as well at the colony level as they did in the laboratory. And then, of course, that leads down the road to regulation, which is we are not a regulatory body. Some parts of USDA are, of course, for pests and pathogens and things. But we have to work with the Environmental Protection Agency and the Food and Drug Administration to make sure to pass all their tests, as it were, to get these things licensed. Dr. Judy Chen has been a colleague in the lab for many years, and has historically worked with honeybee viruses. She's also an expert now in Nosema disease, and has a great team of folks there. One highlight from this year in her group was a postdoctoral researcher, Matthew Herman, was studying these compounds called cyclodextrin. And if you Google those up, they're used for all sorts of things. They're actually in food stability products and things like that. They're not toxic to humans. They're sort of widely used, and their strength is it's kind of a complex molecule, and it forms these sort of wells or cavities that can bind stuff. And the first goal here was that it would some had been known to bind pesticides. So looking at these as yet another strategy for mitigating pesticides in bees, and they did so. They actually sorry for the terrible slides for Sunday night. I'm sorry. You can look at the recorded part and hopefully review them at your leisure. But not for every pesticide, but for some of them. Here, chlorpyrifos, which is a strong insecticide, having these cyclodextrin sort of binding agents in the bees did help with bee survivorship in the face of pesticides. So it was a case where, basically mopping up the really bad chemicals showed some promise. A surprise, though, in the middle of all this is that the bees with these treatments also showed much lower virus levels, and that actually turned out to be stronger. Let's see if I can, stronger than the result of the pesticides. So on the right here, these compounds much reduced the virus loads, and especially the interactions. We think that pesticides kind of impact the immune response of bees and thereby favor viruses, and these compounds actually acted in both ways. They maybe bound to the chemical, but they also seem to directly reduce virus levels. So that's a work in progress, but it looks really promising, perhaps, as something to try in field colonies. And these projects were supported by some folks like Project Apis M and Honeybee Health Coalition, then also the USDA itself and the University of Maryland. I just, one quick blurb, this is the first time I actually advertised this, but it came out this year. If you're interested in sort of other people's research, I've put together a set of essays on honeybee research. Actually started with a great Ohioan, Kim Flodham, when he was editor of Bee Culture, and continuing with Jerry Hayes, who's the current editor. We've been writing a short column, the point of which is just to pull out research papers from researchers across the world and kind of highlight a lot of neat stuff that's going on. And so I've put some of these together into basically a packet of those essays, and it's out there, and I can advertise it because I don't get any royalties from this, otherwise I'd get in big trouble. But if you're interested, that's around on Amazon and elsewhere. Okay, so I just wanted to finish by talking about the Diagnostic Service, and this is again, those of us in the lab doing research have been reminded by the not by Sam, because he's too gentle to remind us of this, but by the prior head of this Diagnostic Service, that it was here first, right? Our laboratory is actually, a remnant, or it's grown from. I guess it's not a remnant. It's got more people from a diagnostic service that started almost at the start of the USDA. Which was started by Abraham Lincoln, believe it or not. And the Diagnostic Service, or the bee health part of the Bureau of Entomology, started in the late 1800s, and it was doing bee diagnostics. Dr. White was a scientist in the USDA downtown, right on the mall, who named the American foul brood agent. in 1908, and we've had that kind of service, especially focused on the two fowl broods, for many years, and that continues as a vibrant branch of the laboratory. And it's now run by Samuel Aban. He was trained for many years by Bart Smith, who many of you know. Bart is happily retired in the area, but Sam is carrying on this service. And this in happier days when we could have visitors. These are some students who came and learned about bee health from Sam. He is a classical bee pathologist, so he does a lot of microscopy. Of course, Nosema counts being the primary disease that you can't really do any other way. And he cultures samples. So these are samples that are just being streaked to identify the agent, if it's EFB. We do it for AFB. We both confirm the species by culturing them, but often also look for antibiotic resistance. So he'll put a little disc on these plates and check to see if they're inhibited by tylosin and oxytetracycline, which is the terramycin active ingredient. This is in past years. Again, about half of the samples we get are from beekeepers and half are from apiary inspectors. And I would swear like 25% come from Ohio, from both groups. So both you and your inspectors are very active participants here, and the diversity of really interesting samples. The adult bee samples are routinely checked for Varroa mites, and they used to be checked for tracheal mites, and we're just not finding them at all. So that's been canceled for the moment. And they will, if there are enough bodies for both Varroa and Nosema, they'll also be checked. We'll get Nosema counts for them. As you might expect, most of these samples, even though they're just remnants of dead outs, often had some sign of Varroa. Some of them well, the average was 14% or 14 mites per 100 bees. One sample had 260 mites per 100 bees. So basically, we know in that case, of course, that a lot of the bees themselves had died off, and the mites were just huddled there wondering what happened to their food source. But we do see a lot of signs that Varroa was the problem in a dead out colony. And again, your groups and yourselves, you have, that's not a surprise to you. That's bred into beekeepers now. Many of them had Nosema, some with incredibly high spore counts. And again, the average of 60% of them was over 1 million spores per bee. And that's thought to be a sort of a threshold where, Nosema is not going to desperately bring down a colony or even an individual bee, but at 1 million spores per bee, it's affecting their energetics and their ability to really function well. These are for 2018. We did 6 samples for tracheal mites, I think just for the heck of it. And none of them were positive. Right now, the only place that seems to be churning out any tracheal mites is in the South, I think in Arkansas. And but there's so few that we just don't even sort of pretend to check for this anymore. Brood samples are quite different. So we get, again, a lot of samples from your inspectors and everyone's inspectors and from beekeepers themselves. We only get bad news, so we only get comb or scale when the beekeeper or inspector thinks something went badly. So these numbers are kind of tempered with, this is a non-random set of bees from the US. A bunch of them did have AFB confirmed, and an increasing but still not terribly high fraction of those had some resistance to terramycin. Terramycin's been around since the mid-50s, and it's hanging in there for amazingly. And tylosin does not yet show signs of resistance, or the agent doesn't seem to be resistant to that. But EFB, so had 30% of these kind of crummy brood samples came in positive for EFB in 2018. We don't test those for antibiotic sensitivity. And again, all of your well, the recommendation for you with antibiotics is certainly to follow the rule of the land, which is the rule of Ohio, in use of antibiotics for either of these, and your own economics, if given that you have to bring in a veterinarian at this point. So 30% plus 16, so that's 46% of these terrible looking brood samples had one of the known brood diseases, and over half did not. So we're still puzzled with, you know, a lot of brood disease that's not caused by those two agents. And it could be viral in some cases, it could be cold stress, it could be a nutritional stress. And the fact is, most of the time we just don't know. We haven't really resolved these odd cases yet. Three things we don't do. One is virus testing as a routine matter. We do have virus diagnostics that we can do, but it's pretty pricey, and so we can't do it for every sample coming in. And same with pesticides. We would love to test for pesticides, but it's at this point it's too expensive to do here, and there are other places doing both of these commercially. We are not currently checking for different races of honeybees or different subspecies. And I know you've had some real tragedies, real sad cases of defensive bees in Ohio, and we have too. We've had sporadic that way. So that the word is out on that. I know you all are having good discussions on that. I don't know where that will go, but I had a call last week with some colleagues, and we reminded ourselves basically that you know we've tried to get a new and improved diagnostic for Africanized bees, and there's a need to do this. So I don't know that it will be our lab. I think it's more likely the USDA group in Baton Rouge will take this on, and they seemed willing. But we hope to have a test for that works well fairly soon. Okay, just very briefly, I just wanted to highlight in the bottom right, if you do want to send samples, go ahead and do so. We can't do the adult bees right now. That might change in the next few weeks, but at the moment, we're still at like 30% capacity at the lab. So we can do brood samples. If you have anything, well, I say first off, I'd call your inspector, because your inspectors know best, both what you're allowed and able to do, and they just have seen this so many times, so they can advise you best on them. But if you or if they want you know, another opinion or a confirmation in some cases, certainly you can send your brood samples to us right now, and Sam will process those. He comes in, I think 3 days a week, and runs through samples, because we know we don't want to be part of the cause of an increase in either foul brood, but certainly not American foul brood right now. So we do some of these samples. All right, so that's all I wanted to talk about. I'm going to stop sharing.
If one hive has been affected by pesticides, does that mean that
they are all affected? Yes and no. If it's I mean, you know, how bees recruit to particular patches of flowers, and there are cases, of course, where a colony might be just zeroed in on a recently sprayed set of flowers, a crop, generally a crop or a horticultural patch, and they might get it worse than their others. But yeah, I would say pesticide effects tend to be pretty equal across an apiary, if not a region even, just depending on the activity of crop protection activity nearby. Yeah, I'd say most of the time you could assume that it would affect the whole apiary at least.
Okay. Sherry's got a question. It says, isn't deformed wing virus from excessive mites?
Yes, good question. So deforming virus can be transmitted even from queens to eggs and from worker to worker, but the super spreaders, the bees that keep it just sort of circulating in the colony, the ones who are coughing on everyone, as it were, are usually had a mite parasitize them when they were developing, and they came out with that mite, the effects of that mite, which is about maybe 1,000-fold higher level of virus than the average bee. So other bees might have it, but the ones that we think are kind of the carriers of it, yeah, usually did have a mite during their development that helped get them there.
All right. Dwight's got a question. Any comments on Randy Oliver's research into sustained exposure on OA, oxalic acid, mixed with glycerin in shop rags to treat for Varroa mite?
Sure, yeah. And Randy also, as he'll fully admit, kind of followed some work in Argentina and elsewhere. As he would also fully admit, it's not quite there yet, and it's not labeled and legally available for use. But it has shown promise. So yeah, so in his own work and some work in elsewhere, we did our sort of bit role in that. We did some residue analyses of honey after treating with shop towels, and we're pleasantly surprised that the residue, it didn't look like it enhanced residue there. The practical output of that is probably we won't see these extended release products available soon, but Randy started a healthy discussion on that. But there's motion and a likelihood of sort of further expanding the use of oxalic acid in the summer in general. So I think we found that it's and others have found that it's the fear of it getting into sort of tainting honey, let alone making it unsafe for us, is fortunately smaller than it could have been. So I think I think as a strategy, as a label to treat with the glycerin extender, say, it's not quite there yet, or it's not strong enough to be licensed and put out there. But we use oxalic as a dribble treatment and are big fans of it for several years. So I think it's a great strategy for mites, but I wouldn't necessarily use it with the glycerin extender, extended relief using glycerin just yet until that gets kind of churns through a bit more.
Okay. Ronald has a question. I've been told not to use chemicals. He's supposed to shake powdered sugar on each frame twice, at least a week apart. Is powdered sugar effective?
Oh boy, I wish it was. Yeah, because it seems, it does get bees to groom and it's good for them, right? It's sugar. But the work, I guess and there's probably more recently, but the best work I've seen on this was from Jamie Ellis's lab in Florida, that it just didn't quantitatively reduce mites. So yeah, so I wouldn't say it's effective on mite numbers, unfortunately.
All right. Marcin has a question. What's the best way to send in brood samples? So if you could cover, mostly because I'm an inspector, I understand not putting the comb into a small bag where it's going to get ripped or crushed. Mm-hmm. So if you could go over just the highlights of that.
Yeah. Well, Sam is, and the inspectors are pretty economical folks too, and it costs a lot for them or for beekeepers to ship these sort of priority mail chunks of comb. So they often will use a cotton tip ear swab and actually send those swabs in sandwich bags, and it works. So if you don't need someone to kind of look at the pattern of whatever disease or event you're seeing in your comb, but just want them to put the samples under the microscope, yeah, the cheapest way, and the Postal Service will probably not like me for this, but you can send it in a business envelope, right? It's these sort of cotton swabs, maybe 3 or 4, maybe in their own individual bag if they're distinct colonies, or 2 per colony in bags. And we get a lot of those, and they seem to work, right? I think they do what they need to do, which is that Sam will kind of douse, kind of suspend those in a little more liquid and look under the scope or plate them. So you don't need to send a whole section of comb necessarily if you want, if you suspect one of the 2 bacteria disease.
All right. Andrew's got a question. How accurate are the instant foul brood tests compared to a lab?
Oh, good question. We did the EFB ones because they were the newer ones on the block, and we actually worked with Megan Milbreth, who's up at Michigan State, on a project she's doing with EFB, and they work surprisingly well. They were 95% concordant with Sam's diagnosis, and we also have a genetic test that those of us doing genetics think has to be the best of the best, right? It's DNA, and they all worked about the same. So they were all equally positive and no false positives. I would say, actually, I think Sam's, sort of the microscopy full route was probably a bit more accurate, but only by a couple percentage points. We did 130 some samples from her, and I think the kits maybe missed 2 at the most. So they did really well.
And I appreciate that, and being an inspector, because I use them myself.
Yeah.
We have a question. Is it true that mites cannot become resistant to formic acid?
Oh, and I've heard that with oxalic too. So it is a physical, whether it's abrasion or affecting breathing, just the causticity, it's a physical attack or insult on the mite. So they won't probably get a cell level resistance to it. The most they could do to get a little more resistant would be to change their life histories by not spending as much time roaming around on adult bees and getting right back into a brood cell, but that won't help them obviously in times without brood. So I'd say, there's the sense is they won't evolve resistance to the organic acids anytime soon. So, which is good news.
A question from John regarding the food stress issue. He's not asking for an endorsement of any specific issue or any specific product, but not all of us know the specific pollen and nectar flows. Supplemental shortings coming in the scientific identification. What is a good supplement to give to the bees to assist in their nutrition?
Oh boy. I would pass the buck maybe to Miguel or to that. I mean, I think, you can't maintain bees on supplements year round or half of the year even, but the ones for palatability and probably ultimately good for the bees, if there's a pollen-based supplement and the pollen used is local, right? We're not likely to introduce a new parasite or disease, then I'd say the ones that are more pollen based are more likely to be a balanced diet for the bees than ones that are based on other protein sources. But Miguel might differ from me on that. He's actually trying some liquid amino acid diets to kind of boost things along, but with the understanding that we don't know enough about all the fatty acids and other components of pollen to really make an artificial diet for bees. We're not there. The science isn't there, and the bees aren't ready for that. They need pollen at some level. So I'd guess at the moment, the more pollen-based ones, as long as that pollen doesn't carry viruses or Nosema, for example.
Thank you. And I'm not sure who it is because it's a device. It says, I've heard powdered sugar is hard for bees to process, and then it can damage their digestive system. Any fact to
I
that?
would believe that. I mean, again, if you're doing mite counts with powdered sugar, it's certainly easier on them than alcohol or ether or even water. But yeah, I wouldn't supplement at all, with powdered sugar. It's either the anti-caking agent or that, or it's just hard. They do clean it up and eat it from each other, but yeah, I've never heard of it per se being used to as a long-term sugar feed or carbohydrate feed.
All right. One from Dwight. Any recent research into small hive beetles, and what would you recommend for the best methods to address?
Oh boy, yeah. There's a lot of research right now. There's a scientist in Florida with USDA looking at pheromones, and there's this yeast that they carry, and actually it is this sliming agent when you get a breakdown from small hive beetle. The yeast is called Kodamaya, and it releases smells that beetles find super, super attractive. And that's when they come in for the kill to a colony that's already got, the starts of a heavy infestation. And so there's some work with baits doing that. I just don't think there's any that are attached to a trap yet or being heavily used. We tried the drier fabrics approach. We don't have a lot of small hive beetle in general, but if the bees are willing to do some work and chew up this stuff a little and make it more fluffy, I guess, they do seem to trap some beetles, but not like in the hundreds. It's just a few. So I would say the promise right now is these sorts of pheromone lures, but I don't think there are any commercially out there. And then, putting your colonies on a hard substrate. Our campus has a bunch of abandoned buildings and parking lots, so most of our apiaries for the beetles and for ticks and things like that are set up on pretty long slabs of concrete, like tens of feet, so long enough that a wandering larva would really have to work to find a home to dig in. So that may help a little bit. I don't know. We haven't used the Guard Star, these sort of drench treatments. Arguably, if you're just getting persistent return of the same beetles, that would probably help too. But the main thing is to keep your hives full with bees and kind of let them push them out as much as possible, or reduce the hive size so they can protect with what they have left.
All right. I get this question as far as being an inspector too. This one says, so would a queen with deformed wing virus from an egg only, or would the bees that raise her be a factor also?
That's a good question. I would almost say, I know you never really find Varroa with queens, but to actually get deformed wings from that virus on the workers, which we've studied a lot more, there needs to be a mite involved. You just can't get these sort of, it's a 10 million virus copies kind of that cause the kind of the threshold for getting deformed wings. So I would almost guess, I'd be really curious to see queens with deformed wings because I haven't seen them, but I would guess it might be another developmental anomaly that caused that, unless somehow there was a mite involved in the raising of that queen.
And my own question on top of that, if the mite load got high enough in the virus load, wouldn't that also cause black queen cell virus?
Black queen cell virus it's not moved around by mites, and it seems steady. Like all the bee informed surveys suggest, it's decoupled. It doesn't reflect mite levels.
Okay.
So yeah, it's a weird one. It reflects some sort of stress on bees, and it gives back some stress, but we still don't really know how it's moved around in the hive. And it's a puzzle. Same with chronic bee paralysis virus, which isn't really linked to mites, and it's not quite as common in the U.S., but it's it's a big problem in Europe, and the people are still trying to figure out, how these things get passed around between the bees.
All right. That's all the questions we have. I want to thank Dr. Jay Evans. I mean, that caught my attention, and a great PowerPoint. A couple little glitches, but we will fix that in post so everything will look perfect.
Okay.
I want to thank him for taking the time out this evening and working with me up to this point to have him online. Next, the end of the month on August 23rd, we have Diane Wolf with high magnification, low cost of macro garden photography. So she can take a honeybee and blow it up so you can count the individual hairs. And you don't have to have the $3,000-$4,000 cameras. You can do it on a budget with a lot of the things, little Kodak throwaway cameras and things like that she's got a lot of good tips and tricks. I want to thank everybody for coming on tonight. We do have an exit poll, so I could ask you to fill this out. And I'll launch this one here just 1 second. Did you learn anything tonight from Dr. Jay Evans? And how do you feel the length of the presentation was? Do you feel more comfortable with using Beltsville Laboratory? And I do say this as an inspector also, please go to the site, because if you're going to send in chunks of comb, there's different ramifications and certifications they need to have the comb in a box, not in a flat envelope where it's going to be crushed and some things. So please use that. And if all else, I mean, if you're in question, ask for your county inspector. Ohio has one in almost every county. I think there's 4 counties that do not. So everybody, thank you for taking the time tonight. Thank you, Mr. Evans.
Thank you. No, thanks for the great questions, and keep up the good work out there.
We will.
Ohio's a good state for beekeeping, for sure. Good beekeepers.
Thank you very much.
All right. Take care. Yeah.